Hey everyone, hope you're all having a great day. I just want to say thank you for joining our webinar on TOC analysis in pharmaceutical manufacturing, non-specific methods and their importance. Introducing myself, I'm Caleb Harvey, Junior Product Manager here at Texwipe, and it's a privilege to introduce today's speaker, Tony Saavedra, Life Sciences Product Manager at Veolia Water Technologies and Solutions. Tony holds his bachelor's in electronic engineering technology from ECPI University, and he has his MBA from Colorado State University. At Veolia, he is focused on Siever's TOC software and instrumentation. Tony began his tenure with Siever's product line as part of GE Analytical Instruments in 2011 in the Field Service Organization, and later held responsibility for leading the North America Technical Service Team, where he oversaw technical support, factory service, and refurbishment process operations. Prior to his positions with Siever's products, Tony served 10 years in the U.S. Navy, where he supervised flight inspections and performed quality assurance inspections and maintenance of complex electronic radar and communication suites. Today, Tony will share his expertise on TOC analysis, its critical role in pharmaceutical manufacturing, and why non-specific methods are key to ensuring water purity and regulatory compliance. So with that, Tony, the floor is yours.
Thanks, Caleb. Good day, everyone. Welcome to our webinar on total organic carbon, or TOC, analysis in pharmaceutical manufacturing, non-specific methods and their importance. So as Caleb mentioned, I've been with the Siever's brand for over 13 years now, starting out in field service, moving my way up into tech support, but in the whole time working with applications, providing guidance with that for customers. In my current role, I am a product manager for the M-Series product for the Siever's brand. So today we'll be discussing the critical role of TOC analysis in pharmaceutical manufacturing, focusing on non-specific methods and their significance. We'll also touch upon cleaning validation and provide examples to illustrate these concepts. So let's jump into it. We'll first start by understanding what TOC is and its importance in pharma. One of the applications for TOC in pharma is cleaning validation, where TOC plays an important role as a non-specific method and is used to determine the overall cleanliness of a system versus specific methods. We'll look at different analysis methods to measure TOC to help understand which is the right method for your application. So let's understand what TOC is, right? So water is indeed the lifeblood of humanity, and its purity is crucial, especially in pharma. Within a seemingly clear glass of water, there may be a hidden world of organic compounds that can impact the product quality and safety. TOC measures organic compounds in water sample, identifying all visible and invisible organic compounds dissolved in water. This is particularly important in pharmaceutical manufacturing, where water purity directly affects the quality and safety of medications. So what is TOC? TOC is an analytical measurement that quantifies the amount of carbon present in a water sample. It's a non-specific sum parameter that captures all organic molecules containing carbon, from simple compounds like methanol to complex substances such as large pharmaceutical molecules. Specifically, TOC is the difference between inorganic carbon and total carbon in a sample. The result gives us a number typically expressed in parts per million or parts per billion. It's important to note that TOC doesn't identify specific compounds. Instead, it provides an overall indication of organic contamination or purity in a water sample, making it an invaluable tool in pharmaceutical manufacturing. So where does TOC come from? Starting off with natural sources, TOC is all around us in our everyday lives. This includes decaying plant and animal matter, right? So as organisms decompose in water bodies, they release organic compounds. This process varies seasonally, with higher TOC levels often observed during the fall. Algae and microorganisms, right? So algae blooms and the life cycles of various microorganisms contribute to TOC. As these organisms grow, reproduce, and die, they release organic compounds into water. Humic or fulvic acids, these complex organic compounds can leach into water bodies and potentially turn them brown. And lastly, tannins. These are found in plant leaves and bark and can dissolve into water, often giving them like a yellowish or brownish tint to it. Another source of TOC is produced by us, right, humans. The first is industrial effluents. The wastewater from industrial processes often contains organic compounds like solvents, oils, and detergents. A few more include agricultural runoff. Those pesticides and fertilizers from farms can wash into water bodies, influencing algae growth and increasing the TOC. Municipal wastewater, treated effluent from wastewater plants still contain organic matter. And then landfills. Water that travels through landfills can pick up dissolved organic compounds. And if it enters into the groundwater or surface water, it can significantly increase TOC levels. Moving into the pharmaceutical industry, there are a few key areas that can contribute to TOC. So ion exchange resins, biofilm formation, and activated carbon filters all contribute to TOC, whether it's through resin degradation or the release of organic byproducts. In addition to those three, APIs, excipients, even cleaning agents and sanitizers and equipment lubricants can also influence TOC number simply by entering the water system during a manufacturing or cleaning process. Another place to consider is within laboratory and sampling practices. Not only improper sampling techniques, but plastic materials and airborne contamination can affect TOC results. Some examples include inadequate flushing, hand contact, and cross-contamination during sampling. From there, the sample containers and tubing can leach contaminants into the sample or absorb and release organic compounds. All while the air potentially contains VOC, dust particles, and aerosols. Now that we've discussed some of the sources of TOC, let's discuss why it's important for pharmaceutical manufacturing. You can see the list on the screen, so let's go through them. First, by identifying where TOC enters the water system, you can monitor critical points and implement targeted treatment strategies. This proactive approach allows for maintaining consistent water quality, which is essential for pharmaceutical production. Next, with real-time TOC testing, when a TOC spike occurs, you can quickly investigate and take corrective actions, minimizing disruptions to operations. Also, a comprehensive grasp of TOC sources also ensures compliance with regulatory standards, including good manufacturing practices, GMP. This compliance is intrinsically linked to the next bullet point on the screen, product quality and safety. After all, TOC can significantly impact drug stability, efficacy, and safety. By identifying contamination points, trending data for out of specifications, and reducing production delays, TOC analysis can help lead to a more cost-effective operation and help mitigate unwanted risk. All of this knowledge feeds into a continuous improvement efforts. The last bullet. As more is learned about TOC sources, companies can refine their water system design, operation, and maintenance, staying ahead of potential issues. Now that we've talked about TOC sources and why it's important to monitor TOC in your facility, let's talk about the different methods available to you. Overall, there are three primary methods are used in pharmaceutical TOC analysis: membrane conductometric, non-dispersive infrared detection, or NDIR, and then direct conductometric detection. These methods are all slightly different So we'll cover the advantages and disadvantages of each. In short, TOC is calculated from the difference between the total and inorganic carbon within a sample. Organic carbon is oxidized by analytical tools, then converted to carbon dioxide or CO2. And then after oxidation, the resulting CO2 can be detected by the tools. Different analysis methods have their strengths and suitabilities depending on specific applications. Let's focus on membrane conductive metrics detection. A highly regarded method for TOC analysis in pharmaceutical water systems. This video demonstrates how this method works, and I'll walk through as we watch. So membrane conductive metric detection operates on a unique principle. First, the water sample is oxidized, typically using a UV light or chemical oxidizer. This process converts all organic carbon in the sample to CO2. Next, the CO2 produced from the oxidation is separated from the liquid sample. This is done using a semi-permeable membrane that only allows CO2 gas to pass through, as you can see in the video. From here, the CO2 passes through the membrane, is absorbed into a pure water stream. We call it our DI water side. This absorption causes a change in conductivity in the DI side. The change in conductivity is directly proportional to the amount of CO2 present, which in turn corresponds to the amount of organic carbon in the original sample. So now that we've seen how this method measures TOC, let's look at the advantages and disadvantages. There are several key advantages to this method, as you can see from the video alone. We see that high specificity is an advantage because this method is measuring only CO2. This eliminates the possibility of interference from other ions or compounds. Membrane conductive metric can also detect very low levels of TOC, often down to the parts per billion or parts per trillion in specific applications, which is crucial for ultra-pure water used in pharmaceutical manufacturing. Because of the semi-permeable membrane, the conductivity sensor is protected, therefore requiring minimal maintenance. Real-time analysis is another advantage, which is highly valuable for process control in pharmaceutical water systems. And finally, the last two advantages are robustness and compliance. So membrane conductive metric is less affected by variations in the sample composition compared to other methods, making it reliable across different types of pharmaceutical waters. It's also fully compliant, meeting requirements set by pharmacopoeias and regulatory bodies for TOC analysis, such as USP 643, the monograph for TOC, and then 1225 for validation of compendial methods. However, it's important to note that while highly effective, this method does have some limitations. One is the initial cost. So the equipment for this method can be a little bit more expensive up front compared to some of the other TOC methods. And then the sample range. So due to the nature of the analysis and the ability to maintain stable calibration over a year, the upper range is around 50 parts per million. Despite these minor drawbacks, membrane conductive metric detection remains a go-to method for many pharmaceutical companies due to its accuracy, reliability, and suitability for continuous monitoring and high-purity water systems. Now let's turn our attention to NDIR detection. Another important method for TOC analysis in pharmaceutical water systems. I don't have a nice video for this one, but let's walk through it. The NDIR starts in a similar way to other methods. The sample enters and is first oxidized to convert all organic carbon to CO2. Then there's an infrared light source that emits a beam at a specific wavelength that is only CO2 can absorb. The CO2 from the sample absorbs some of this infrared light, and then the amount of light absorbed is proportional to the concentration of CO2 present. By measuring the amount of infrared light that reaches the detector and is not absorbed, the instrument can determine the CO2 concentration and consequently the TOC in the original sample. This method also has some advantages and limitations, so let's start with the advantages. CO2 has a unique absorption spectrum in the infrared range, making this method highly specific for CO2 detection. In addition to being highly specific, NDAR can operate across a broad range of concentrations from low ppb to high ppm TOC levels. It's also generally stable and requires less frequent calibrations compared to other methods, but you may need additional calibration ranges, right? So starting out from very low, maybe 0 to 50 parts per million, and then a 50 parts per million up to 1,000 parts per million. The NDR method has the ability to provide quick results, which can be beneficial for real-time or near real-time monitoring as well. However, NDR detection does have some limitations. For example, water vapor can interfere with the accuracy and the sensitivity of the analysis. This is because water vapor also absorbs infrared light, potentially leading to overestimation of TOC levels. Another limitation is that NDR detectors can also be affected by temperature fluctuations and physical shocks, requiring careful control of the analytical environment. Also, while this method does have a wide dynamic range, it may not be as sensitive as other methods. Sample preparation and possible drift are two other limitations to keep in mind. In order to minimize water vapor interferences, samples often need to be dried or dehumidified before analysis, which can add complexity to the process. And over time, the infrared source and detector can experience a drift, necessitating regular calibrations to maintain its accuracy. Finally, let's look at direct conductometric detection. This method also operates on a different principle. It measures the conductivity of the water sample before and after oxidation. As you'll see in this short video, the process involves the sample entering the sensor where initial conductivity measurement is made. Then the sample is then oxidized. Only with the UV lamp, no reagents are added, and a final conductivity measurement is taken, and the amount of TOC is calculated based on the conductivity difference. So let's move to the next slide to see the advantages and limitations of this method. This is a straightforward method, right? We can see the advantages are quickly. Direct conductometric is reagent-free, simplifying the process and reducing operational costs. Because of this, results can be obtained quickly, making it more suitable for maybe real-time monitoring instead of maybe a real-time test. So if you're just trending information. And as we saw in the video, it's rather simple and generally less complex than other TOC analysis methods, which leads to it being a cost-effective method. Both initial equipment and ongoing operational costs are typically lower than other methods. It does have several limitations, though. So direct conductometric is more of a sensor than a true analyzer due to frequent interferences from other ions or compounds that affect conductivity. Because of this method, it cannot be validated per USP 1225. And then direct conductometric also doesn't directly measure inorganic carbon, only providing a differential measurement, which can lead to inaccuracies. It assumes all conductivity increase is from CO2, which isn't always accurate, especially with non-ionic organic compounds. These assumptions also make calibration challenging, and it may hold true across different types of organic compounds. Finally, direct conductometric also has limited sensitivity and may not be sensitive enough for ultra-low levels of TOC required in some pharmaceutical applications. In pharmaceutical manufacturing, this method is typically used for trend monitoring or preliminary screening, or in conjunction with more accurate, validatable methods. It's essential to understand its limitation and interpret results cautiously, especially when dealing with water systems that may contain non-ionic organic compounds where compliance with pharmaceutical standards is required. So let's look at some considerations. So let's talk about what's the right TOC analysis method for you. Right? So we've covered a lot of ground here today, so I want to break this down into something practical you can use. When you're deciding on a TOC method, there are several factors you need to keep in mind. So let's walk through them. First would be accuracy and precision. So if you're dealing with ultra low, ultra pure UPW water, ultra pure water for critical applications where you need highest accuracy, the membrane conductive metric detection is usually your best bet. NDIR is also good, offering a solid accuracy across the wide range, but may not be as sensitive at very low levels. Now, direct conduct metric, well, it's less accurate and precise, so mainly useful for just trend monitoring. Next, think about regulatory compliance, and this is a big one in the industry. Both membrane conductor metric and NDIR methods can be validated according to USP 1225. But here's a key point. Direct conduct metric cannot be validated to the standard, which limits its use in regulated pharmaceutical environments. Then there's the detection range. For those ultra-low TOC levels, membrane conductor metric really shines. NDI offers a wide dynamic range, so it's good for low to high concentrations. And then direct conduct metric, it might struggle with very low TOC levels. We also need to talk about interferences. So membrane conductor metric is least affected by these, followed by NDIR. But there are ways to deal with that, right? So direct conduct metric is most susceptible to interferences, especially from non-ionic compounds. Now, what if you need continuous monitoring? All three methods can do this, but direct conduct metric is often chosen this for simplicity and lower cost in its application. Lastly, think about ease of use. Direct conduct metric is usually the simplest to operate. NDIR and membrane conductor metric might need more specialized knowledge, but they give you more analytical power. So to sum it up, if you're working on critical pharmaceutical applications that need highest accuracy and have to meet pharmaceutical standards, membrane conductor metric detection is often the way to go. If you need a good balance of accuracy and a wide range of detection, right? So NDIR might be an excellent choice for you. If you're more focused on trend monitoring or preliminary screening or less critical applications where absolute accuracy isn't crucial, direct conduct metric is suitable. But here's an important point. In many pharmaceutical settings, you might end up using a combination of methods. For example, you might use direct conduct metric for continuous monitoring, but then periodically verify with membrane conductor metric or NDIR methods. At the end of the day, the right choice really depends on your specific applications, what regulations you need to meet, and your operational constraints. In my advice, always consult with analytical experts and really think about your unique needs when making this decision. Let's transition a little bit to talk about the role of TOC in cleaning validation, which is a critical aspect of pharmaceutical manufacturing. Cleaning validation is the process of ensuring that equipment and production areas are consistently cleaned to prevent cross-contamination between products. TOC analysis plays a crucial role in this process as we take a closer look over the next few slides. Let's start addressing a common question in cleaning validation. Should you use swabs or is final rinse analysis sufficient? The answer, as with many things in our industry, is it really depends. Swab sampling involves physically wiping surfaces with a swab to collect residues. You typically use this method when you're dealing with equipment that is hard to rinse thoroughly. Think about equipment with crevices or rough surfaces or complex angles, right? You may also use this method when contaminants are not easily soluble in a rinsing solution. Another reason for to use this method is to validate the cleaning of specific worst-case locations. These are typically hard-to-clean areas that represent the greatest challenge in your cleaning process. And then finally, you may use this method because the regulatory expectations or guidelines specifically call for swabbing. On the other hand, final rinse analysis involves collecting and testing the last rinse solution used in the cleaning process. This method is often sufficient when equipment is easily rinsed with smooth surfaces and simple angles. You may also use it if the contaminants are readily soluble in rinsing solution, and you can demonstrate that the rinse solution has contacted all of the surface of the equipment. It may also make sense to use this method if the equipment is too large or complex for comprehensive swab sampling to be practical. In many cases, a combination of both methods provides the most comprehensive validation. For example, you might use a final rinse for your routine monitoring with periodic swab sampling of critical areas. When deciding between swab or rinse methods, consider the equipment design, product characteristics, right? Is it soluble or not? Your cleaning process, regulatory requirements, and resource availability, as swabbing can be more labor intensive and time consuming. Remember, whichever method you choose needs to be validated. You must demonstrate that your chosen method can reliably detect residues at the levels of concern. Let's discuss why TOC is such a valuable non-specific indicator of water quality in pharmaceutical manufacturing. There are several key points to consider here. First is a comprehensive detection. TOC analysis provides a broad overview of organic contamination in water. It doesn't just look for one specific compound. Instead, it detects all organic carbon present. Next, it's sensitivity to various contaminants. So TOC is highly sensitive to many types of organic compounds. It can detect contaminants at very low levels, often in the parts per billion range, making it an ideal tool for monitoring high purity water systems. TOC can be an early warning system, right? Because it's comprehensive in nature and high sensitivity. TOC often serves as an early warning system for water quality issues. An unexpected rinse in TOC levels can alert you to potential problems before they become critical. Real-time results. Compared to many specific analytical methods, TOC analysis is relatively simple and quick. Most modern TOC analyzers can provide results in minutes, allowing for near real-time monitoring of water quality. TOC is also complementary to other tests. So while TOC is powerful on its own, it's also complementary to other water quality tests. For example, while conductivity measures ionic contaminants, TOC covers non-ionic organic contaminants, and then used together, these tests provide more complete picture of water quality. Then trend and monitoring. So TOC is excellent for monitoring trends in water quality over time. By tracking TOC levels, you can identify gradual changes that might indicate developing issues in your water system, such as biofilm formation or degradation of purification media. And then process validation. TOC plays a crucial role in validating water purification process. It can help demonstrate that your purification system is consistently producing water that meets the required quality standards. This is essential for regulatory compliance and ensuring that the safety and efficacy of pharmaceutical product is met. Now that we've covered TOC as a non-specific indicator, let's take a closer look at a non-specific versus specific monitoring. All right, let's talk about an important topic in cleaning validation, which is that specific versus non-specific monitoring, with a focus on TOC analysis. This is crucial when you're designing your cleaning validation strategy. First, let's define our terms. All right, so what is specific monitoring? That's when you're testing for a particular compound or a contaminant. As you can see in the image to the left, Think HPLC to detect residues of a specific active ingredient. Non-specific monitoring, on the other hand, casts a wider net. It detects a broad range of substances without identifying them individually, as you can see in the image on the right. That's where TOC analysis comes in. Now let's look at the pros and cons of each approach. For specific monitoring, what's the good? It gives you definitive identification of contaminants. It can be highly sensitive to target compounds, and it's great when you need to prove the absence of a particular substance. What's not so good? It's often very time-consuming and expensive. You need to separate methods for different contaminants, and as you might miss unexpected contaminants, such as excipients, cleaning agents, and don't forget about the degradation of products. HPLC might nail that API detection, but it can miss everything else. Now, for non-specific monitoring with TOC, on the plus side, it detects a wide range of organic contaminants. It's generally faster and more cost-effective. It can catch unexpected organic contamination and is suitable for routine monitoring. Here's where TOC really shines. It doesn't care if it's detecting an API, an excipient, a cleaning agent, or a degradation of product. If it's organic carbon, TOC will find it. So while your HPLC method is a laser focused on that one API, TOC is giving you a comprehensive picture of all organic residues. So what's the downside? Right? It doesn't identify any specific contaminants. It might be overly sensitive, picking up harmless organic residues, and it can't detect inorganic contaminants. In practice, many companies use both approaches. And it's worth noting that some regulatory bodies like the FDA have shown support for non-specific methods like TOC in cleaning validation, but you need to justify your approach and limits. While TOC is a non-specific method, it offers many advantages in cleaning validation. It's not a complete solution on its own. The most robust cleaning validation program often uses both specific and non-specific monitoring methods, taking advantage of the strengths of each approach. TOC gives you that broad coverage, catching APIs, excipients, cleaning reagents, degradants, all in one go, while specific methods can provide a detailed information when you need it. So let's walk through a real-world example of TOC analysis in cleaning validation, or hypothetical. I think this will really help illustrate the process and highlight some key points you need to consider. So picture this. We're validating the cleaning process for a tablet press. This press has been used to manufacture a product containing ibuprofen. Here's how we might tackle this using TOC analysis. First, we need to establish our acceptance criteria. Based on the toxicity of ibuprofen and the surface area of your equipment, let's say we've calculated our TOC limit is 10 parts per million. Next, let's take a baseline measurement. We measure the TOC during our cleaning solution before we start. Let's say it's 0.5 ppm or 500 ppb. This gives us a starting point. Now we perform our standard cleaning procedure on the tablet press. After cleaning, we take our samples. We're using two methods here. So we take the swabs from the critical, hard to clean areas of the press, then we collect the final rinse sample as well. For the sample prep, we extract the residues from the swabs into a known volume of high purity water. The rinse sample is ready to go as is. Now comes the analysis. We run our samples through the TOC analyzer. Let's say we get a result from the dye cavity swab. It's 8 ppm from the punch swab and 5 ppm on our final rinse. From the dye cavity is about 8 ppm, from the punch swab is 5 ppm, and our final rinse is 3 ppm. So what do these results tell us? Well, first off, all of them are under our 10 ppm limit. That's the good news. We see that the dye cavity has the highest residue at 8 ppm, and that makes sense, right? Typically, it's a hard to clean area. Our final rinse is lower at 3 ppm, which is typical. It represents an average of all of the surfaces. Now, as part of our validation, we also ran an HPLC analysis for the ibuprofen on duplicate samples. These show a good correlation with our TOC results, which gives us confidence in our TOC method. Based on all of this, we can conclude that our cleaning validation process is effective. All of our TOC levels are below acceptance criteria. But then let me highlight a few key points here. Now, remember, TOC isn't just detecting our ibuprofen. It's picking up any organic residues that includes excipients and cleaning agents as well. Notice how the swab results are higher than the rinse samples. That's typical. Swab gives us more of a worst case picture. We're using both swab and rinse samples. This gives us more of a comprehensive view of our cleaning effectiveness. And that correlation with HPLC, well, that's crucial for validating our TOC method. Now, in practice, we repeat this process multiple times to ensure it's reproducible. We'd also challenge our process by intentionally dirtying the equipment to prove our cleaning can handle the worst case scenarios. This example, I believe, really shows how TOC can give us a quick, comprehensive check of the cleaning effectiveness. It's sensitive enough to catch low levels of residue, but broad enough to pick up unexpected contaminants that a specific method might miss. Let's recap what we've covered today about total organic carbon analysis in pharmaceutical manufacturing. We started with the basics. So TOC is our key indicator for organic contamination in water systems. We explored how it can enter our process from various sources, potentially affecting product quality. We then examined different TOC analysis methods: membrane conduction metric, NDIR, and direct conduct metric. Each has its place, and choosing the right one depends on your specific needs. A significant part of our discussion was focused on cleaning validation. We've seen how TOC as a non-specific indicator can enhance our approach, potentially improving efficiency while maintaining rigorous standards. Implementation is crucial, right? So method development, validation, and understanding matrix effects. These aren't just technical details, but essential steps for ensuring reliable results. And in today's regulatory environment, robust data management is more important than ever. A key takeaway: TOC analysis is a vital component of quality control in pharmaceutical manufacturing. It's not just about clean water or clean equipment. It's about ensuring product integrity and patient safety. I hope this webinar has given you valuable insights to TOC analysis and its importance in our industry. Consider how you might apply these concepts to enhance your own quality control strategies. Thank you for your time today. I'd like to open it up to any questions.
Thank you, Tony, for that presentation. Very insightful. I felt like I got my own personal lecture right there. It was perfect. But yeah, no, like you said, let's just open the floor for questions. Hey everyone. So yeah, no, just feel free to leave any questions down. I see a question right here. Sean asks, which TOC analysis method would work best for cleaning validation?
Yeah, great question. So to answer this question, we really need to think about the advantages and disadvantages for the different testing methodologies, right? Because cleaning validation contains ionic and non-ionic species. You also must be able to quantify each one of the actual individual measurements. So the direct conduct metric or DC technology probably isn't the right choice for you because you need to be able to actually detect carbon, right? And not just assume that carbon is coming from carbon dioxide, which also, which can lead to interferences, right? Both NDIR and the membrane conductive metric technology would be ideal. So you must decide at what sensitivity level, right? Because some of the rinse state samples that you're looking at can be very low in TOC levels, and where membrane conductive metric really shines.
Got you. And we do have another question here. Can you automate cleaning validation?
Yes, you can, right? So we do see a lot of people moving to the automation side, right? So you're decentralizing the lab. So instead of a lab-based test, you're moving to either app-line or to online analysis. But you really need to understand your process, right? Whether that rinse state sample is really representative of your swab samples, right? So you can move away from the swabs. If you can't get away from the swabs, right, you won't be able to fully automate the process. Then you also need to think about what technology you're going to use, which one is appropriate for an online measurement to fully automate that process and integrate it into your facilities.
Got you. And you mentioned interpreting the results from direct contaminant cautiously. If there is an out of specification, can you use the data to help out with an out of specification?
And so DC technology, right? So when you look at how it's validated, per USP 643, it's only a limit test, right? So it only tells you if it's above 500 ppb or below 500 ppb. So you can't really use that information quantifiably. You just know it's either above or below that specific limit. So this is where the MC technology, membrane conductive metric, actually is used. It can be really used for both process control as well as process understanding to help aid during an out of specification.
Got you. We also have another question here. Alfredo asks, what swab do you recommend for TOC analysis? And is it possible to reuse the vials when you carry the samples, or is it not recommended?
Ooh, it's a tough one. So you need to have a swab that is validated, right? You need to make sure that it doesn't contain any TOCs. It can gather the appropriate what's the word I'm looking for here? Drawing one of those blanks. But it must be able to put it into sample. As for vial reuse, don't typically recommend that, right? You'll need to prove that vial, because once you put a sample into a vial or swab into a vial, right? Because then you're introducing contamination, right? So you need to be able to have either a way to clean that vial out or just use a whole new vial to actually be safe. A lot of vendors, including ourselves, we actually sell certified less than 10 ppb TOC vials. So you can actually just reuse those vials knowing that there's no contamination in those vials and then just discard after you use.
Perfect. And we have one more question here. Do TOC samples have to be submitted in water?
Yes, TOC analysis requires an aqueous solution. That being said, many compounds which are considered hard to solubilize, even in water, can be used to can be detected using TOC with a suitable method development, right? So introducing heat, agitation, or acid to a sample can often increase the solubility of a compound so that TOC analysis is readily detected for the compound. For example, TOC recovers have been demonstrated for difficult to oxidize and insoluble compounds like lidocaine or starch, right? As part of a method development, it's essential to demonstrate the proper recovery of these compounds by writing protocols to demonstrate the linearity of the recovery.
Perfect. Well, let's add, do we have any more questions in the chat? And if we don't, I just want to say thank you to everyone that attended. Don't want to take too much of your time today. I will be sending out a follow-up email, but if you have any personal questions that you have about the webinar, maybe getting a recording, or any specific questions for Tony, feel free to email me. I can go ahead and get those answered for you, or I can kind of or direct you to Tony just if you have any more specific questions for him. But again, I will be sending up a follow-up email just to everybody saying thank you for attending. And yeah, no, thank you all for coming, and I hope you all have a good rest of your day.
Thank you, everyone.